Hello and welcome to Biology Lesson 2.07 on proteins. And you'll notice right away that I didn't put polymers of proteins because proteins in and of itself means polymers of our monomer, which were amino acids. So we said back in our monomer introduction, monomers, we have 20 amino acids. And I'm going to kind of put a box around this because these are my nucleic acids and we're not going to do this. So we're going to have to do this on the next page. I can tell right now I'm going to run out of room. We talked about three polymers in carbohydrates made from glucose. In lipids, we talked about two different categories of fatty acid chains. We mentioned the phospholipid. Proteins is another game. We're in a completely different ballpark. There are some estimates that you have 400,000 proteins in your body. One estimate says 400,000. Let's put that in perspective. The English language, if you get a Webster's dictionary, has 170,000 words. And in your body, you have 400,000 proteins. How is that possible? How do we go from having 4 or 5 polymers and carbs and a couple of polymers and lipids to having 400,000 proteins? Your body is all about proteins. Your cells are all about making proteins. Well, we have a picture of a protein down here. And it turns out that we start off with proteins. So if we're going to build a protein, let's talk about building a protein. We start off with an amino acid chain. All right, I'm going to pick a different color here. So just like we did with the glucose, I'm going to do amino acid. And I'm going to put a connection between there. And remember, I have 20 different amino acids, and so I can have quite a lot of variety in how I string these together. This is called my primary structure. We're going to fast forward a little bit. There's actually different levels of folding this guy up. So we go through a secondary, we go through a tertiary. But what happens is I have a string of hundreds of thousands of amino acids, and it goes from this to this. Look at that shape. Isn't that unique? This is called our, and I'm not going to test you on this, but you might just be interested. This is called our quaternary structure. And it has a very, very distinct shape. So everything about proteins ties back to their shape. Proteins are, the whole purpose for a protein is to get a compound that's going to have a specific shape. So they are, what's the word I want to pick? Identified, or their characteristics, everything about them, their identity is linked to their shape. Now this is a 2D picture. If we could hold a 3D model, we would see it's pretty darn specific. But, and I believe this is hemoglobin, which carries around oxygen on your red blood cells. I don't know, I could be wrong about that. So if I had to kind of give this a shape, it kind of looks like this to me. If I could give it an overall shape, if we're going to simplify it. And I want to look at one example. By the way, we get from here to here by folding. So we do lots of folding. An analogy is that I always give is proteins are a lot like origami. If you've never heard of origami before, it's called Japanese paper folding. And they always start off with a simple structure, a sheet of paper. In this case, we start off with a string of amino acids. But an origamist can make these incredible designs by folding the paper. So they can make paper swans, they can make flowers, they can do all of these incredible structures just by folding their paper differently. And that's what happens in our body. We take these primary structures and we fold them into these proteins. So this is a string of amino acids. This is actually a protein. An example of this are something called enzymes. Enzymes are a group of proteins, and an enzyme is a protein that causes a reaction to speed up. When I worked as a chemist, we had to come up with coatings that would protect car parts from rust. Now, if we had to test those parts by setting them outside and waiting to see if they would rust, that would not have been very beneficial because it can take years and years and years for a screw or a bolt to rust. So we have what was called a salt spray cabinet. We will put them in this cabinet. Let me check, make sure my power's turned on. We would put them in this cabinet, and it would get sprayed with a mist of salt, and the cabinet would be hot, and it would speed up the corrosion process. And so we could test our corrosion resistance by speeding it up. That's what enzymes do in our body. If you had to digest your food without enzymes, it would take maybe a year to digest a hamburger. That's not going to work out for you very well. So how do these enzymes work? Let's go with light green. So I'm going to draw an enzyme protein, and we're going to take the one we've got here. So this is my enzyme. Let's say this is a digestive enzyme. And let's pretend like it's even got a name. We're going to call this lactase. Lactase is the enzyme that breaks down lactose, and lactose, if you're lactose intolerant, you can't drink milk. So lactase is the enzyme that breaks down milk, lactose or milk. We have an area on the enzyme that will bind with a substance, and this is called the active site. The substance it binds to, the substance that binds to an enzyme is called the substrate. And let's draw my lactose as blue. So let's say that lactose has this shape. It's like a mushroom. Let's make it shaped like a mushroom. That's a complete fiction, but it's going to prove our point. So this is my substrate. If I don't have lactase, this guy's going to float around forever. And actually, for a lot of us, this is the case. I am lactose intolerant, and I don't really have enough of this enzyme. And so if I drink a glass of milk, I will throw it up because my body can't break it down and it sees it as something that's dangerous. If you do have enough lactase, this is what happens. So from here, the lactose binds to the lactase at the active site, like this, and they form what's called a complex. So when they're bound together, it's called a complex. And the enzyme changes the shape of the lactase, and it cracks it in half. And what happens then at the end is My enzyme isn't changed. The enzyme's the same way as it was when I started, but I have these two halves of my lactase, and now my body can use this. My body can take this and break it down the rest of the way, but my enzyme remains unchanged. This process, when you have a substance that speeds something up but isn't changed by the reaction, it's called catalyzing. So speeding up a reaction, this is called catalyze a reaction. Now, you can buy pills that have this enzyme in it. I don't think they work that great, but some people say that it helps them out. And that's called Lactaid. It basically has the lactase enzyme in it. All right, so what are our takeaways? There are far more proteins than any other category of molecule. 400,000, that's a bunch. To build a protein, we take a long string of monomers, amino acids, and we fold them up, and our proteins have this unique shape, and everything about a protein is tied to its shape. The example we gave was lactase. Lactase is an enzyme that catalyzes the breakdown of milk, or lactose. We have a substrate which binds to the enzyme at the active site. The enzyme then causes the change in the substance that it's bound to when it's in a complex, and the substance leaves broken. We can do this in reverse too. Some enzymes cause things to connect together, so it can go the other direction. If it weren't for enzymes, our body would basically starve to death because we have to have digestive enzymes to help break down our food, enzymes to do lots of other things. And this is just one category. Remember, we said proteins also help with communication, transportation, anything in your body you need done, it's probably a protein. All right, that is our video for proteins. Thanks for watching and have a great day.